This is Corey. Lately, he's been feeling rather sluggish and has been mysteriously losing weight. When going out for drinks with friends, by the end of the night he noticed that he had some pain in his lymph nodes. At the insistence of his friends, he went to the doctor. And upon running some diagnostic tests, they found that he is one of the 8,500 people per year in the US who are diagnosed with Hodgkin's lymphoma. Luckily, this cancer has a high survival rate and there are multiple treatment options. However, one of the most affordable and effective options, vinblastine, has a limited supply. So what can be done about improving the availability of these treatments? And what new developments are being implemented to combat this? Hi folks, my name is Cole and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at a microbial supply chain of chemotherapeutic drugs. So hang around with me throughout this whole video to get all of the relevant background information so we can dive into some exciting experimental results. There's also more information for you in the description below. Now, plants produce some of the most potent human therapeutics and have been used for millennia to treat illnesses. Monoterpene indole alkaloids, MIAs, are a structurally diverse family of complex plant secondary metabolites with many medicinal properties, and they are found mainly in gentianal plants. Now, streptozidine is the precursor for over 3,000 MIAs. It is also the precursor for vinblastine, which is what we're going to be focusing on today. Vinblastine is used to treat a variety of cancers, like Hodgkin's lymphoma, testicular, breast, and lung cancer. It can also be used to treat fibromatosis and germ cell tumors. It is so effective at fighting cancers that vinblastine and its analog vincristine are included in the World Health Organization's list of essential medicines. They exemplify some of the few established MIA therapeutics on the market, with an annual market size of more than $1 billion. Now, if vinblastine is so rare, why not just synthesize it chemically? Unfortunately, MIAs in general are very difficult to chemically synthesize from basic substrates. We get vinblastine from Catharanthus roseus, more commonly known as Madagascar periwinkle. To extract just 1 gram of this medicine, it requires 500 kilograms of dried leaves. This purification process also yields many other plant molecules, including vindoline and catharanthine, the precursors for vinblastine. Now, these two can be coupled together enzymatically with horseradish peroxidases, chemically using iron, or through excitation with lasers, allowing for the production of additional vinblastine from the C. roseus leaves. Cancer is the result of rapid, uncontrolled cell growth arising from cellular mutations. Vinblastine works by preventing the cancer cells from separating into 2 new cells. It does this by blocking the microtubule structures that divide the cell in the final steps of mitosis. Recently, in 2019, the full 31-step biosynthetic pathway of vinblastine from C. roseus has been clarified. The enzymes responsible for this are localized in at least 5 compartments in C. roseus cells and involve trafficking between different tissues and cell types. Knowing about this is a great step forward, but as with all knowledge, it needs to be applied for us to make full use of it. Baker's yeast has been used as a microbial platform for characterizing newly discovered metabolic genes in plant-specialized metabolism. Yeast is streamlined for the investigation of numerous plant enzymes because it partially shares its primary metabolism with plants. It is also able to be efficiently transformed with plant genes, has the capacity for post-translational modification of proteins, and has limited endogenous specialized metabolism. These processes are aided primarily through the use of plant cytochrome P450 enzymes, which speed up metabolic interactions. Now, yeast has been used extensively in the production of useful materials. For example, insulin used in diabetes treatments is produced by simply inserting the human gene for insulin into the genomes of yeast. Furthermore, researchers from Stanford University have engineered yeast to produce opiate-like chemicals, and others still have used yeast to produce cannabinoids, terpenes, and alkaloids to treat cancer, pain, malaria, and Parkinson's disease. Now, these processes start out at a tiny scale, growing yeast in just milliliters of liquid. Using bioreactors, the process is then scaled up to thousands of liters, enabling production of medicines at an industrial scale, providing people around the world with greater access to medicines at decreased cost. Now, I want to take a moment and really highlight why using yeast as bioreactors for chemotherapeutics is so important. As I mentioned before, vinblastine is listed as essential by the World Health Organization, and there is a scarcity of it worldwide. The natural resources required to create vinblastine are immense, and the current process to manufacture it is inefficient. Thus, by using scalable fermentation, refactoring complex plant biosynthetic pathways of human therapeutics in microbial hosts could promise more access to plant natural products and new-to-nature derivatives. If you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper that we're focusing on today. This paper is called A Microbial Supply Chain for Production of the Anti-Cancer Drug Vinblastine by Zhang et al. From the Technical University of Denmark and Kongens Lyngby, Denmark. In this paper, the authors demonstrate the de novo microbial biosynthesis of vindoline and catharanthine using highly engineered yeast and the subsequent in vitro coupling to make vinblastine. To refactor the vinblastine pathways in yeast, the authors divided the pathway into 3 modules on the basis of substrate availability, and used generative versions of modified yeast, which include the expression of C. roseus genes to help support the pathway's enzymatic activity with cytochrome P450. Now, the initial strictosidine strain, MIA-AU, was designed on the basis of a previously reported 12-step de novo strictosidine strain. This yielded a total of 5.5 micrograms per liter of strictosidine, which is not all that much. To increase the yield, the authors prevented some of the byproduct generation by knocking out some genes, and the swapping of target promoters when bottlenecks were identified via supplementing different precursors in the growth medium. This involves substituting gene analogs from multiple different plant species to optimize production. To produce strictosidine de novo from glucose and tryptophan, the authors modified intrinsic yeast genes to ensure increased production of GPP, tryptamine, and enhanced ethanol metabolism. The authors further tested different yeast media and found that YPD media could be used to create 25 milligrams per liter of strictosidine, 4,500 times more than the initial concentration. Having optimized strictosidine production from its constituent parts, or de novo, the authors next sought to express the gateway enzyme SGD to progress the pathway. This was measured indirectly via a downstream intermediate. And no matter how hard they tried, the authors could not get the C. roseus plant variants of SGD gene to work. So they looked at these cellular locations and found that C. roseus SGD was in the cytoplasm, while Revolfia serpentina SGD protein, which did work, was in the nucleus, where it needs to be to function. To see why this might be happening, the authors looked at R. serpentina SGD protein to determine the essential parts for function. They modified it into all available conformations and found that the 3rd domain is required for activity, while the 3rd and 4th domain together produce the most measurable analyte. Nevertheless, to refactor the tibercinine/catharanthine module from C. roseus in yeast, the authors integrated all of the necessary genes for catharanthine synthesis, including the R. serpentina SGD gene, and found that this resulted in 9 micrograms per liter of tibercinine and 7 micrograms per liter of catharanthine. In parallel, the authors also inserted all of the required genes for vindoline generation, alongside the final gene, PRX1, which is meant to synthesize vinblastine from vindoline and catharanthine. This yielded 1 milligram per liter of vindoline, but no actual vinblastine. Using targeted metabolite analysis to look for potential bottlenecks or issues with the pathway, the authors found that the P450 enzyme T3O needed to be optimized, while confocal analysis showed that PRX1 was not being displayed. Frustratingly, nothing the authors could do would get PRX1 to express, so they moved on without it. When the catharanthine and vindoline modules were combined together, this yielded 4 micrograms per liter of vindoline and 44 micrograms per liter of catharanthine. With the multiple parts now in working order, the authors next sought to integrate everything all together. They first combined the strictosidine and tibercinine pathways, followed by the vindoline pathway, and finally adding in the gene necessary for catharanthine synthesis alongside some other gene edits. By adding in some extra copies of genes in the tibercinine pathway, the authors were able to get the yeast to produce 5.5 micrograms per liter of vindoline and just over 69 micrograms per liter of catharanthine. Now, to get even more catharanthine and vindoline, the authors grew these yeast in sequential bioreactors, giving the authors a grand total of 91 micrograms per liter of catharanthine and 13 micrograms per liter of vindoline after 11 days of growing, doubling and tripling the yield of catharanthine and vindoline respectively compared to just the 2 modules alone. Now, even though the authors could not synthesize vinblastine in the cells, they still wanted to synthesize vinblastine from their biofactories. The authors found that after purifying synthesized catharanthine and vindoline, they were able to successfully use iron 3 chemical coupling to effectively generate 24 micrograms per liter of vinblastine. Now, to quickly summarize everything all together, the authors utilized yeast as a factory unit to create vinblastine precursors. They did this by inserting all the genes required for the 30-step vindoline/catharanthine pathway. First, they broke up the pathway into modules and optimized them for biochemical synthesis. By optimizing the strictosidine module from the beginning components, they were first able to increase the amount of strictosidine created by over 4,000 times. They then inserted genes which utilized strictosidine to create the vinblastine precursors vindoline and catharanthine. This is the limit of what the authors could do in their cells because, try as they might, they could not get vinblastine to be synthesized within their system. However, by purifying the precursors from fermentation broths and through the use of chemical coupling, the authors could synthesize vinblastine, thus providing the framework for using yeast as effective biofactories. Not only do I think that the genetic modification of yeast for creating chemotherapeutics is exciting to investigate and learn about, it is also significant in a broader context. This information is significant because it firstly illustrates that yeast can withstand extensive gene insertions to recreate complex plant biochemical reactions, showing that microbial biofactories can be used for essential anti-cancer medicines. By refining the process and scaling up manufacturing, these engineered yeast could reduce the amount of resources needed to create chemotherapeutic medicines and other derivatives. Now, all science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions myself after reviewing this information. Getting yeast cells to fully synthesize vindoline and catharanthine all the way from tryptophan and glucose is no small feat. But I think the next obvious question is whether it would be possible to make vinblastine inside of the cells. It should be, right? Perhaps by using another yeast strain as an intermediate through development, or by finding or creating a PRX1 gene that will synthesize vindoline and catharanthine together. Along the same train of thought, what are the measures and modifications that will have to happen to allow for industrial scale synthesis of these compounds? Because, quite frankly, they're not the same as growing up small volumes of cells. There's many more factors to consider. Additionally, because strictosidine is the backbone of so many other MIA compounds, how could the initial module be used in conjunction with other potential modules to make multiple plant-based therapeutic compounds? As always, my final question revolves around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about them in the comment section below. Also, let me know if there are any topics that you'd like to hear about in the future. Ultimately, I hope that you learned something, but more importantly, I hope that you enjoyed your time doing so. If you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I'll see you next time.